So, in today's zoology lecture, we are going to see the renal physiology, and this is a study about the kidney and their structure and their function. Let's begin with the introduction part. So, here in this zoology lecture, we have to study about the renal physiology. So, if we go through with the structural analysis of the kidney, that is a very important and it is a retroperitoneal part of our body for refining all the waste material from inside to outside the body. So, this is a major function that has been done by the kidney in our body. And if we see the weight, like how much weight it possesses from our body, that will be like 0.5% of the total body weight. And approx if we see the individually, that is 150 grams in the weight. So it is covered by the renal capsule and renal fascia. So majorly there are two covering which is present on the kidney. That is one is renal capsule and second one is renal fascia. Now, if we see the structure of the kidney in little detail, so you can see here, the kidney is divided into an outer cortex and inner medulla. So this is the outer cortex, okay, and they also have this inner medulla. This is the medulla portion, and this is the outer cortex region. So the medulla is composed of the renal pyramids. Okay, so these renal pyramids they are present inside the medulla, or you can say the medulla of the kidney is totally composed of this renal pyramids. These are have to be separated by the renal column. So these are the renal column. You can see this. Okay, these are the renal pyramids, and these are the renal columns. So they are just separated by this renal columns. The renal pyramids empty urine into the calyces that drain into the renal pelvis. So this is a renal artery, renal pelvis, okay, and this is the renal vein. So here, these whole pyramids they needed to be empty the urine into the calyce. So this calyce can be brought to it to the renal pelvis, and they can help them to excrete out from the body. So from there, urine flows into the urethra and it's transported to the bladder to be stored. So after that, they will be transferred them to the urethra, and then they will be transported to the bladder. So there it can be stored and excrete out when it is needed, or you can say that when the composition of the urine is having more in the quantity, so it is going to be excrete out. Now, this was a general structure that is all about the kidney. Okay, now we have to see their general functions, like what kind of function that has to be performed by this kidney. So, the very first function is regulate the ECF volume. Okay, this ECF volume is a very regulation point or inside our body that has to be very maintained. Regulation of electrolytes regulation done by the kidney. Regulation of arterial blood pressure. So arterial blood pressure should be regulated by kidney only. Regulation of pH. So acid base balance has to be done by the kidney only. Like by adjusting the amount of water, they can help to regulate the pH also. Excretion of waste product, that is a natural work that has to be done by the kidney. Anything which is presently waste inside our body and not no longer going to be used inside, so they have to be excreted out. Endocrinal function, so the renin, erythrosin, active vitamin D, so basically that is called the endocrinal function. This is also done by the kidney inside our body. Last, degradation of hormone. So the hormone which have been used, and now if we wanted to degrade these hormones, so the whole degradation process will be done inside the kidney. Now, if we talk about the functional organization of the kidney. So there are two major parts which contribute the kidney formation. Very first is the renal cortex, and second one is renal medulla. So if we talk about the renal cortex, that is red and dotted in the structure, contain glomerulus. So glomerule will be presenting into the renal cortex, contain proximal tubules, distal convoluted tubules, and cortical collecting ducts. So these are the three major components which have to be present inside the renal cortex. First is proximal tubule, distal convoluted tubules, and cortical collecting ducts. In the renal medulla, they are pale and radial in shape. Renal cortex was red and dotted, but renal medulla they will be pale in color and have to be radial in symmetry. So contain renal pyramids. So the pyramid which we just seen in the diagram, they will be present inside the renal medulla, which contain minor calyces that unite to form major calyces. So each and every pyramid they are having a minor calyces into them, and after the unitation of these small minor calyces, that going to convert into the major calyces. Contain loop of Henle and medullary collecting ducts. So they will be contains loop of Henle and medullary collecting ducts. So basically this medulla is going to have the loop of Henle will be inside them, and they also have the collecting ducts inside them. Now we will see the diagram, a detailed diagram, clear one. This is a renal cortex, okay, and these are the renal medulla that is present in a side. So this renal pyramids, light pinkish, dark pinkish color, and they you should see that they are present like a chambers, and these chambers are interrupted by these renal columns in between them. These are the renal column. These are the renal chambers or pyramids. This is the renal pelvis. This yellow one that is called the renal pelvis, and this yellow scattered version that is called renal papilla. So this renal papilla and renal pelvis they are having a major role in the excretion process. Now this is a renal cortex, the outer version. Medulla is the inner side, and this outer region that is the cortex. Now here you can see the two things: minor calyx and the major calyx. So this is the minor calyx and this is a major calyx. Renal medullas, renal artery, renal veins, urethra, renal papilla. These are always present inside the medulla of renal or renal medulla. Like this is a renal medulla, and this red one that is the renal artery, which basically like all the blood, how the kidney going to help in the blood stimulation. So at that time this renal artery is going to way, or you can say from this renal artery the kidney going to help in the blood function. Next is the renal vein also. The vein and the artery, why they are present, we know the what the major function has to be performed by the artery and veins. Artery follow the oxygenated blood, and the veins they take the deoxygenated blood to the heart. So these whole things, the both kind of the veins, both kind of the glands are present here, and the urethra renal papilla that is also going to be present here, as also the minor calyx will be. These yellow and very elongated thread-like structure is also present here. These are called nephrons. We will take how the nephron will be structurized inside the kidney. Next here, the blood supply of the kidney. So how the kidney is going to help or inside the blood supply, or how the blood supply process happen inside the kidney. So kidney receive its blood through the renal artery. So we just studied this is the renal artery which is present here. So this renal artery, through this renal artery, kidney is going to receive it blood. The renal artery is divided into the anterior and posterior branches. So there are two branches is going to be present inside the renal artery. One is anterior, second one is a posterior. Each one divided into five segmental branches. So there will be each one of this renal artery. So they will going to be divided or segmented in a five regions. What are the five regions here? There can be arcuate, interlobular, efferent, glomerular, if the capillaries, they're afferent or the arterial. So these are the five things in which they can be present. Interlobular, acute, afferent, capillaries or arterioles. So these are the pattern in which they are going to be present inside the kidney. Now the afferent arterioles, they are the pretubular capillaries and vasa recta in the interlobular. If we talk about the vein, they are the interlobular vein of the segmental, renal vein and IVC. So these are the main source of the blood supply inside the kidney. So, or you can say these are the organs which are responsible for all the energy getting or the blood supply inside the kidney. This glomerulus, you can say this is a whole the thing which we studied here. These all, which are the main source for the blood supply in the kidney. They are diagrammatically represented here, like the glomerulus part, glomerulus capsule, this one, and this is the glomerulus. Efferent arteriole, this is that. Okay, afferent arteriole, that is here. Second one, interlobular artery, that is present here this way, a small one here. Proximal convoluted tubule, that is this blue one, sorry, this yellow one here. This that is basically attached at the central with the glomerulus, and arcuate artery and veins, they are present here. Both the artery and veins in the blue and artery in the red. Interlobular artery and vein, these are the interlobular artery and veins, and they are the arcuate artery, and these are the interlobular arteries. Pedubular capillaries, so they are present here, just behind the glomerulus. Distal convoluted tubules, they are for in the passage which going to in the interlobular vein. And this distal convoluted tubules, they have to be present in a last manner. This one, the yellow one, that is called a collecting duct. So this collecting duct, all the things which are coming from these all, they will be going to collect in this main collecting duct. So these are interlocular veins and the distal convoluted tubules, ascending limbs or loop of Henle. This is called loop of Henle. Okay, ascending limb and descending limbs. One is going to be increased, one is going to be reduced. So this kind of ascending and descending proceeding of limb, that will be known as loop of Henle. So this loop of Henle and the collecting duct, that is the two major important things that has to be understood by this diagram. Now, this is the whole diagram of a one kidney. You can see these are the intralobular veins. The blue one, they are the intralobular veins. This is the red one, these are the intralobular artery. This is a renal column, okay, in center. This the white matrix type thing, that is a major calyx. Papilla of or this orange brownish content, that is a cortex. This one, that small covering of white, that is the minor calyx. This red portion here, that is a medulla, medullary renal pyramids. Okay, in this you can see it here, or in this you will be can be able to see at this place. So basically, all the parts they are essential. In this diagram, you can able to see at what positions they are present inside the kidney. You can also see the arcuate artery, arcuate veins. They are just forming these loops, and here, when they are having in the ascending and descending order, they are going to make the loop of Henle. Lobar artery, you can see this, segmental artery, this, renal artery here, renal vein here. This is important where you can say the whole process will be going to take place in a final duct. At that position, this renal artery and renal veins will be present. Now the most important word here, that is called the nephron. So we have to see what is a nephron, structural and functional unit of the kidney, and how it is going to be work inside that. So it is a structural and functional unit. So in the it is like you can say this is a unit of kidney. So renal corpuscle and renal tubules. So corpuscle and renal tubule they are having two different kind of nephrons inside them. So if we talk about first by the renal corpuscles, so they have to form of the glomerulus 20 to 40 loop of capillaries between afferent and efferent arterioles. So they will be meet up in between the afferent and efferent arteriole and there will be loop of 20 to 40 loops. Now the second one is here the Brownsman capsule. So Bowman's capsule. So which is the blind end of the proximal tubule. So as a blind end of the proximal tubule, that is known as the Bowman capsule. So that's why this is also made up by the nephron. Now, second here, the renal tubules that is formed of the proximal tubules. Okay, here will be forming of the proximal tubules. Intermediate tubule loop of Henle. Okay, second one, intermediate tubule of loop of Henle. Distal tubules. So in the distal tubule, they will be coagulated tubules connecting tubule and collecting to the duct. So these are the process, or you can say how this unit is going to be a structural and functional unit of the kidney by performing these all structures and the functions. Also, you can see in the diagram here, distal convoluted tubules. This is a nephron loop. That's this loop. Proximal convoluted tubules, renal cortex. This whole thing, or combinedly, they will be known as juxtamedullary nephron. So, in with all these structural analysis, with all these function, they will be going to name as a juxtamedullary nephron. These are the proximal convoluted tubules, renal corpus cut, glomerulus capsule, distal corpuscle tubules. So these are going to be made the cortical nephron in a factor way. Next here, in a detailed version of the diagram, you can see the cortical nephrons, okay, present here, and the juxtamedullary nephrons, they are present here. So the two type of nephrons is going to be present in the kidney. One is the cortical nephrons, and second one is a juxtamedullary nephrons. So they both of these kind of nephrons they are present in a renal cortex. You can see in the diagram also they are present. More of their part will be present in the renal cortex, and alternatively they are just binding up, or you can say they are just storing or collecting all the matter inside the collecting duct that is centrally placed in between all these nephrons. So this is glomerulus and this is a blood flow like arteries flowing blood inside, and the disorgan oxygenated blood or with the waste product means they are coming out. Now, this type of nephrons, that are the two types of nephrons here. One is the superficial cortex, and second one is the juxtamedullary. So, what happened in the superficial cortex? That is made up of 85 to 90% of the glomerulus in the outer two to third part of the cortex. So, all the outer part of the cortex, or you can say when the two-third part of the outer cortex, that is glomerulus, will be formed like 85 to 90% with the superficial cortex nephrons. Second, loop on Henle is formed of DLH and ALH. So loop of Henle will be formed here. Loop of Henle mark this bend the junction between outer and inner medulla. So basically, loop of Henle which form they make a bend at the junction point in between the outer and inner medulla portion. Small in size with low capacity to reabsorb salt and water. Their size is small, so that's why their capacity also reduced for the absorption of salt and water. Juxtamedullary, that is about 10 to 15% present in the outer cortex. Glomerulus in the glomerulus in the inner one third of the cortex, like two third is already has been taken by the superficial one. So juxta just going to remain with the one to the third part of the cortex. The loop of Henle is formed of DLH and ALH and thick ALH. So this loop of Henle, in that case, they are just made up of by the DLH and ALH. So here, the this loop of Henle, there will be forming by the thick ALH formation. Loop of Henle makes a bend at the tip of inner medulla. So they are going to make a bend on the tip of inner medulla and large in size with high capacity to reabsorb sodium and water. As their size is very high, or you can say their size is very huge in the juxtamedullary, so that's why their capacity of absorption of sodium and water is also high as comparison to the other particles, or you can say as compared to other nephrons. So these are the type of nephrons in diagram representation. That one that is called the cortical nephrons, juxtamedullary nephrons, okay, efferent arterioles, pretubular capillary network. You can see how the capillary network is working around the nephrons. Interloculi artery glomerus. This the glomerus structure you can see that is formed by the arteries here. Outer stripe, outer medulla, inner stripe, arcuate artery. So this is the outer strip of the outer medulla, and this is the inner strip of arcuate artery. So you can just simply see the two different kind of nephrons here. That is basically divided in the inner medulla and outer medulla of the renal cortex. Now we see the juxtaglomerular apparatus in a little detail. So this is a proximal tubulus. This is a mesogenal cells. That is the Baumann's space. That are the granular cells, efferent articles, vascular smooth muscles. That is a macula densa, efferent articles, glomerulus capillaries, plexus wells, and the marginal cells. So this is a whole combination to form a juxtaglomerular apparatus. Okay, this is not just a nephron. This is a whole apparatus that has been made up by different component and when they are come together. So it is system formed of modified. So this is a generally you can say this is a modified version. How they will be modified here, like the macula densa cells, okay, juxtaglomerular granular cell, extraglomerular mesenteric cell. So these all the three kind of cells have to be performed inside this juxtaglomerular apparatus, because if out of three, even if the one is going to missing, the apparatus cannot work efficiently for their transportation or any work which they have to be performed, like the ion stability or the transfusion, excretion. So for the any kind of information or the working process, this whole apparatus have to be needed to present in that apparatus. Now we will take some cells one by one to see how they are going to affect. So very first here, macula densa cells. So they are the modified tubular epithelial cells. So if we talk about them, so they are the epithelial cell which has to be modified into the tubule-like structures. Present at the transition zone between a thick ALH and DCT. So if we see their position, like at what position they are present inside this whole apparatus, so they will be present in the transitional zone, and where between the thick ALH and DCT. What they do there? They monitor sodium and chloride concentration in the tubular fluid. So their work is to see that the concentration of sodium and chloride ions, they should not be very much inside the fluid, tubular fluid, or not very less. So they have to be present in a equilibrium manner, or you can say they have to be present in an accurate manner to do their function efficiently. Now, juxtaglomerular granular cell. This is the second kind of cell here, which we have to see. So, in this, present in the wall of the afferent arterioles, they have to be present in the wall of afferent arterioles. not in between the transition zone as the Macusa densa present, they were present in the transitional zone. They are present inside the afferent arteriole. They are smooth muscles with epithelioid appearance. So, if we talk about them, they are the smooth muscle-like structure, but their appearance is epithelioid appearance, like the epithelial tissue appearance. They form, store, and secrete renin. So, what is their function? They needed to store the renin. First they do the formation of renin. Then they were going to store the renin, and they were going to secrete the renin if needed. Contain B1 androgenic receptors, so they have also a quality to contain these androgenic receptors, because something if going to wanted to be bind at the surface, so they have the receptor for their binding. So they just help to the binding properties molecules to be bind on the surface of the any nephron apparatus. Act also as a barriero so sometimes also they can be some barriers can be happen when the receptor and molecule function for any molecule, like the signaling or transporting some information from one place to another place. So at that time a couple reaction happen, but in the opposite to the couple reaction there can be a barrier reaction. So that's why in that case they act as a barrier acceptor. So they can even they can accept the barriers which stopping some kind of reaction. They can accept that too. Now extraglomerular messenger cells, what they do? So it is a continuation of the intraglomerular messenger cell. This is a direct contact with the mucosa densa and juxtaglomerular glandular cells. So it is a continuation of intraglomerular messenger cells. So this cell which we just studied is this glomerular granule cell. So they will be going to the continuation in that messenger cells, and they are direct contact with the mucosa densa and juxtaglomerular granular cell. So if they are going to be in the direct contact with the two of these kind of cell, so passing of the thing from one to another place, that will be easily done. So that's why they needed to be in touch with each other. Otherwise, if the distance between any two leaves get long, or it may be left out, so that the problematic condition can be happen even in the collecting of the main duct, or secreting some enzymes, or decaying some hormones. So the very function which have to be done by the kidney, the any one function can be disrupted if they are not present too close. Now there are some stimuli for renin release. So basically, as they cell, they have been stored the renin inside. So there will be some stimuli which going to help to renin to release out. So everything work in our body, they work on the some kind of stimuli. So here the sympathetic stimulation, the very first, which is called on the sympathetic based. So this stimulation, they need sympathetic layers, renal ischemia, that is inside the renal artery, or sometime that renin can be released by the decrease of sodium and chlorine concentration in the tubular fluid. So as in the tubular fluid, our first cells is medulla densa, they needed to be monitor the concentration of sodium and chloride. But if their monitoration can have any kind of decreasement in the sodium and chloride, then there will be, and or you can say that situation will be a stimulus for the renin release. So these are the three things which can be work as a stimuli for renin release. Now, renin angiostensin aldosterone system. So this is whole system which is happen inside or in between the kidney and liver, and some inhibitors which going to be to the angiotensin aldosterone system. So basically, in this we know the basic functionology, that is depend on the three condition between renin, angiostensin, and aldosterone. As a RAS is going to start up and kick up in the liver, they are going to release this angiostensin inside here. Then they will be got the ANG1, and from the lungs they are having the ACE, and also sometimes the ACE inhibitors can be work here. So after the ANG1 formation, there will be this ACE. They will stimulate the ANG1 to form into the ANG2. Now the another thing that is Saturnus that will going to stimulate ANG by the adrenal gland, and they will be converted into the aldosterone. Okay, so that aldosterone in the, you can say, when the blood pressure rise or H2 retention, the water retention take place and BP rise, at that time this aldosterone will be produced inside the nephron. So now this nephron will, they are going to make this process to be collecting in a duct, or they are going to be release the renin. So you can see the whole, the kidney is producing renin, liver is producing angiostensin, whole process will be going to perform the aldosterone. So here renin from kidney, from angiostensin from liver, ACE from lungs, adrenal glands, even the BP and hydrogen from aldosterone. So all these processes are come together just to make a complex of renin, angiostensin, and aldosterone system. So this, you can say, they are collectively working here for their process. Now, what are the inhibitors of the renin release? So, these were the promoters which you studied here. These all are the promoters. Now, we have to see some inhibitors also. So, in the case of inhibitors, this aldosterone, okay, that is the first inhibitor that present here. Arterial neurotropic peptide, that is called ANP, that has to be released by the lungs. All hypernatremia. So this hypernatremia, aldosterone, ANP, and all these can be collectively or individually can do the inhibition of renin release. So if the aldosterone will be going inside the nephron, as you already see it here, if the aldosterone going to be here inside this collecting duct, then kidney stop producing the renin. But if the liver is going to produce an angiostensin, that will going to stimulate the kidney to produce renin. So they are the stimulator and they are the inhibitor. The renin is just, you can say, the intermediate position between the angiostensin and the aldosterone. One is going to stimulate their process to be come out by the kidney, and second one is going to work as a negative feedback mechanism for their production. Now, the function of the renin-angiostensin system. So, what are the functions which have to be performed inside this renin-angiostensin system? So, there are two type of the thing. Very first is called the vasoconstrictions. So, what happened in the case of vasoconstriction? So, the arterial constriction increase the afterload, or you can say the arterial blood pressure. So, in the vasoconstriction, there will be a arterial blood pressure. So, arterial blood pressure that will be happen by the arterial constriction that basically involve their increasement. Second, this vasoconstriction, they can be increase the preload, that is called the venous return. Okay, so this preload can be also happen when the this vasoconstriction increases. Third here, salt and water retention. So salt and water, this function of renin and angiostensin system, they are also helpful for the retention of salt and water from the kidney. Angiostensin 2 and increase the sodium reabsorption from proximal tubule. They also can enhance for the reabsorption of sodium by the proximal tubule. A proximal tubule which has been present in the kidney, so they can be responsible for the reabsorption of sodium again inside the tubule. All stimulates the zona glomerulosa of the adrenal cortex to secrete aldosterone that increase any reabsorption from the still tubule. So here, all stimulate the zona glomerulosa. So this layer of the this glands of the zona glomerulosa of the adrenal cortex. So this have to secrete aldosterone. So they needed to be secrete aldosterone, and they just secrete this aldosterone only when the resin angiostensin system work. So you can say this aldosterone formation, that is a result of these resin angiostensin function, and they will also increase in the sodium reabsorption from distal tubule. Basically, the angiostensin 2, that is going to increase the reabsorption of sodium by the proximal tubule. But here, these aldosterone, they are going to help to increase the sodium reabsorption from the distal tubule. Now, what are the pharmacological inhibitors of RAS, or you can say the renin angiostensin system? So, angiostensin converting enzyme inhibitor decrease the formation of all. So, basically, there are some converting enzymes like the angiostensin 2 blockers, decrease aldosterone release, and B blocker. So some very, you can say the pharmacological is not appropriate, but we can say some reverse in negative feedback mechanism can do their inhibition process very efficiently, and some B blockers, they also have this tendency that they can inhibit the this RAS process inside the body. Now, I think this is all about the kidney, and you have the enough exposure, like what kind of tubules and what kind of nephrons is present inside the kidney. What is the basic function of kidney, and what particular Cano system performing a generation of hormonal control, what kind of arteries are working here, what are their physiological function, at what place they are present. So these all things, I hope these are clear. And now we have to see that you have to go through some questions. So after answering this question, you will be able to know that you got something or not. So where are the kidneys situated? So that's the very first question. What is the approximate length of thickness of kidney? You can answer that. What is the structure that encloses the kidney? Okay, so the covering of kidney. The second slide, I told you that. Which of the following organ is associated entirely in the right kidney? So that organ that is situated near the kidney of right side. Which of the following statement is not true? So one of these statement that is not true about the kidney. Which structure allow the entry of blood vessels? So in which lymph vessels and nerve to enter kidney? So in which there is a one which going to allow the blood vessel or lymph vessel nerve to enter the kidney. Match the following, like there are some terms: cortex, medulla, capsule, and major calyx. So you just have major calyx and minor calyx. So you just have to match with them, like which surround the kidney, contain renal pyramids, outside the renal pyramids, form from the merging of. So these you can match on the basis of their adaptability. What is the function of kidney? So I think that is the simplest question among them. The renal pelvis leaves the kidney as. So if they just talk about the renal pelvis, so how they will be leaving the kidney. What is the approximate weight of the kidney? I already told what was the approximate weight. That can be differ from species to species, but that is a 0.5% of our whole body, and one of them you can found a very closely approximate weight. Now, still, if you, I think it's all about the kidney. We go through it, but still, if you guys have any problem, you can go through these references. These are going to help you to understand more about the kidney physiological function, their parts, how they can be utilized in the body. So this is all for today. I think you get it as I wanted you to be. Okay, class. If you have any queries or if you don't understand anything, just ask me whenever you are free. Thank you. That's all for today.